name is Lynette. I work as a specialist biomedical scientist in the UK. I have 20 years experience working in cellular pathology. I've worked in 7 NHS trusts, and I feel that now is the right time to discuss the basics and some advanced points within cellular pathology. So this is a series of shorts. We're going to try and keep it to probably between 15 to 20 minutes so they're easily digestible. These shorts will be produced in English as it's my first language. I hope to put a translation at least in English and Spanish subtitles, and I want everybody to know that even though my first language is English, everybody is welcome here, irrespective of your first language. So please feel welcome and thank you for being here. So we'll discuss histology today as a whole. We'll discuss the value of histology, how it's used in diagnostics. We'll also discuss some specialist techniques we use, and then also the value of these specialist techniques. And then we'll just have a quick introduction to hematoxylin and eosin. So histology is the study of cellular material, specifically tissues. Histology means histo is him, and ology is a study of anything, so it's actually a study of the human form, effectively. We also have cytology. Cyto is a study of cells, so cells, and then ology is a study of cells. Another word to consider is pathology, and that's the study of disease process and disease tissue. So we'll use histopathological technique to discover issues with regard to pathology within cellular material. We work within the constraints of the Human Tissue Act using a license to hold and work with human tissue. I want you also to know that I don't hold a Home Office license. I have no opinion when it comes to animal model, animal tissue or animal diagnostics. If you want any information with regard to that, please go to another source. So this will be just a short introduction into cellular pathology and cellular pathology technique. So just what is the value of histology? So we provide information with regard to cellular material for the requesting clinician, our specialist scientist or our pathologist will provide reports for the requesting clinician in order to provide a diagnosis for a patient. The tissues we receive come in all sizes, from small fine needle core biopsies to large surgical resections taken in theatres. The source of our material is usually a hospital or hospital group, a GP practice or practices, and then also private clinics and private hospitals that also provide health services. The requesting clinician should be responsible for counselling and informing the patient with regard to the tests that we're performing and the implication of the results of the test we may inform. The samples we receive are in specialised specimen receptacles. The specimen receptacles should bear the patient identifier label and are received with a requesting clinician's signed form as to which tests are required by the requesting clinician. When we receive the specimen into the laboratory, we receive the pot, which is labelled with a specific laboratory number, which is used by our laboratory information system to track this specimen through our process. When a specimen is described macroscopically by eye, the patient identifiers are checked and the specimen is described in colour, appearance and measurement in usually millimetres. The specimen is recorded on the laboratory LIMS system and is placed in a labelled cassette which holds the specimen to be passed for processing later. The forms and the cassettes go through the lab together so we can track the quality of each stage of processing. The processing is performed by a processor. The specimens all are cassetted and placed in a receptacle which pumps hot formalin initially onto the specimens, and then through a series of graded alcohols that remove water from the specimens, then a clearing agent called xylene, which is miscible with the final stage, which is molten paraffin wax. This molten paraffin wax is at 60 degrees centigrade and is useful to us because it supports the tissue. So invariably a lot of routine laboratories use paraffin wax to support their tissues in the wax blocks. Molten wax is good because it enables us to manipulate the tissue and set the tissue in a particular orientation to demonstrate all aspects of the tissue specimen. Once the paraffin wax is set at room temperature, these cassettes can be popped out of the moulds and then placed with the forms again, so we know exactly which tests we're going to perform. The specimens are cut on a specialised machine called the microtome. The microtome provides very thin, probably about 4 microns, thickness of tissue that can be placed on a clean microscope slide. These clean microscope slides are transparent, as too is the tissue. These tissues are then placed on a staining machine routinely, and the routine stain of choice within the UK and internationally is the hematoxin and eosin stain. This stain will demonstrate specific tissue elements to include nuclear detail and cytoplasmic detail within the cells. As a standard, every histology and cellular pathology request has a H&E with the initial processing of the tissue. The slides are stained, dried, and then mounted with a glue and a cover slip. This glue has the same refractive index as the glass, so with the stain preparation underneath, if you shine a light through using a microscope, you can see the specific tissue and cellular elements clearly with a good resolution. So these preparations are quality controlled, QC'd, and they are submitted to the reporting specialist scientist or pathologist. The reporting specialist scientist or pathologist will form an opinion based on the H&E preparation presented and may ask for ancillary tests. So these are other tests that demonstrate things that may be required to be demonstrated for a complete diagnosis of the tissue. Some sites do perform a Masson trichrome stain with the hematoxin and eosin, and that's also submitted with the hematoxin and eosin at the initial stage. And then there are some specialist tissues that are required to have a panel of ancillary tests produced initially at the same stage as the initial preparation, and this is done routinely for those specialist tissues. Once all of the ancillary testing is complete, the specialist reporting scientist or pathologist will form an opinion and a diagnostic opinion based on the preparation seen and the tissue seen. This will form a report which is written and submitted to the requesting clinician. The report and a lot of other information around the patient's diagnosis is discussed in the multidisciplinary team meeting, or MDT, which is performed weekly. These MDTs have oncologists, radiologists, medical clinicians, surgeons, pathologists, specialist reporting scientists present, and together these specialists will provide opinions of the patient's prognosis, diagnosis and treatment plan. A timescale will also be written, and this treatment or time frame and schedule will be presented to the patient and their families. The value of having all of the specialist team together present means that at that moment, hopefully all the information required for treatment and diagnosis of that patient is done at that time, and these MDTs, as I said, are done weekly and usually based on a certain speciality. So all of the speciality teams for a certain discipline, for example urology, they will all be together, so you have the experts that you require at that one time to provide an accurate treatment plan for a patient. So the value of what we do in regards to special stain and hematoxylin and eosin stain is they should tell us information about the tissue on the microscope slide. These things include the cell type present, the tissue type present, if there's any infectious agents or any features of infection present. Also, they should give us clues with regard to the general state of the tissue and if there is any pathology present. So the state of the tissue could range from completely normal tissue, which in fact would be a negative result, to any inflammation repair, pre-cancer and cancer. And that would obviously be at the more positive side of a result. The hematoxylin and eosin stain is the first contact routine stain performed, and this will be discussed in the next video. And that's everything for now. Thank you very much for listening. Generally, if there's any questions anybody needs answers to, I'll try and answer them if you put them in the comments below. I'll try and use references. I'll try and link any useful published resources that I find to support any opinions I have. If I don't know the opinion, I'll try and find a reputable source for a reference. If any person has a specific training need or anything specific to themselves, please talk with your local training officer or training team leader. Thank you for listening. To be a good biomedical scientist, specialist biomedical scientist, medical laboratory assistant or associate practitioner, you just need determination, a commitment to best practice, a commitment to the protocol locally, and a willingness to work in a large team professionally and produce professional standard diagnostic specimens. So as long as you've got that, I haven't met anybody in my lifetime that can't produce sections and preparations of a diagnostic standard. So thank you. I wish you all energy to complete anything you require to do for your studies or career.